Welcome to the deep dive. Today, we're plunging into the world of fish diseases. We'll be looking at common bacterial infections.
Yeah, things affecting farmed and wild fish.
Exactly. And then we'll tackle a pretty notorious parasite, marine white spot disease.
It's a really critical area, when you think about it. About a third of the world's food actually comes from aquaculture now.
A third, wow.
Yeah. So when disease hits, the economic impact for farmers can be huge. And it spills over. It affects wild populations too.
Right. We've got a lot of research here, and it's clear just how diverse these problems are. We're talking bacteria, parasites.
It's a range of pathogens, yeah. Each with its own way of causing trouble for fish health.
So our mission today is to kind of cut through that complexity.
Exactly. Pull out the key stuff for you.
Yeah.
What causes them, the signs to look for, how they spread.
And crucially, how we can prevent and control them.
That's the goal.
Okay, so let's dive into the bacterial side first. Bacteria are just, well, everywhere in water, right?
They are. But the intensification of fish farming, that's where things can get tricky.
How so?
Well, you put a lot of fish close together, they get stressed. It's like us in a crowded space,
Yeah, makes sense.
That stress lowers their immunity, makes them way more susceptible to bacteria that might otherwise be fine.
And then it just spreads rapidly in those conditions.
Like wildfire, yeah.
And the consequences, they can be pretty devastating, can't they? High mortality, slower growth.
Slower growth even affects the quality of the fish meat itself. It's serious stuff.
What kinds of things push it towards an outbreak?
Our sources point to a few key factors. Poor diet, for one, subpar farming practices, environmental shifts like temperature or oxygen,
Okay.
just general weakness in the fish, maybe introducing new fish that carry something. It all adds up.
So understand the specific bacteria is key.
Absolutely. You need to know the agent, its biochemistry, how it moves through the epizootiology.
Yeah.
That's vital for effective management.
It's kind of amazing. Something like 125 species from 34 families linked to infections?
Yeah, though many aquatic bacteria are harmless, of course. But those 125 or so are the ones we worry about.
Mostly gram-negative rods, it says here.
Predominantly, yes. Some gram-positive, a few acid-fast ones, too.
And there's even a human health angle.
There is. Fish can carry bacteria that can infect people.
Yeah.
Usually through eating raw or undercooked fish, or sometimes just handling them.
That's definitely something to keep in mind. And controlling these outbreaks in water must be incredibly difficult.
It's a huge challenge. You can't easily watch every fish, and like we said, disease spreads so fast in water.
Right.
Okay, so let's get into specifics. Gram-negative bacterial diseases first. Flavobacteriosis.
Yeah, that covers a few things, doesn't it? Cold water disease, gill disease, columnaris.
Exactly. Caused by Flavobacterium species, gram-negative, anaerobic, non-mogul rods.
So they don't need oxygen and can't really move on their own.
Correct. The main ones are F. psychrophyllum, F. branchiophyllum, and F. columnar.
Let's start with F. psychrophyllum. Causes rainbow trout fry syndrome, cold water disease. What are the signs?
Okay, so you'd look for fish being off their food, listless, maybe bulging eyes. That's exophthalmia.
Exophthalmia, right.
Eroded fins and skin. White patches are common. Sometimes a swollen belly with fluid, pale gills.
And it's mainly a cold water issue.
Yeah, temperate regions affect salmonids mostly, but some others too. Usually hits juvenile fish when the water's around 12 degrees C or lower.
How does it spread?
Horizontally,
Huh.
often from infected dead fish left in the water. So removing mortalities quickly is really, really important.
Got it. Okay, what about F. branchiophyllum? Bacterial gill disease? Sounds like it targets breathing.
That's exactly right. It goes for the gills. Fish might stop eating, get sluggish. You might see them gasping at the surface.
Gasping.
Yeah, trying to get oxygen. And sometimes they swim really stiffly, almost upright. They call it soldier-like swimming.
Soldier-like. Wow, that's quite a visual.
It is. This one's been known since the 1920s. More common in farmed fish. Spreads horizontally, too. Outbreaks often pop up in spring or early summer as water warms up.
And it attaches quickly to the gills.
Very quickly, yeah.
Okay, the last one in this group. F. columnar. Columnaris disease. Had a few names over the years.
It has. Shows how our understanding evolves. With this one, look for lethargy again. Lots of appetite. Fin rot.
Fin rot, okay.
Fish often hang near the surface. It can cause acute or chronic infections, gills, skin, fins. Sometimes get that classic saddleback lesion.
Saddleback?
Yeah, a lesion on their back. In warmer water fish, tropicals, it can look like fuzz around the mouth fungus or cotton wool disease.
Where's this found?
Pretty much worldwide in freshwater fish, farmed and wild. Younger fish are more susceptible. Spreads horizontally. Handling injuries make them more prone to it.
It makes warmer water.
Yeah, severity increases above 20 degrees Celsius.
Okay, let's move to Edwardsiella, another gram-negative group, big in tropical fish farming.
Seems to be, yeah. Several species cause disease, but E. tarda and E. ictori are the main players.
E. tarda causes Edwardsiella septicemia, also called red disease in eels.
Right, and that lovely name, emphysematous putrefactive disease of catfish.
Doesn't sound pleasant at all. What are the signs there?
Loss of color, weird fluid buildup, bleeding points, little nodules forming, cloudy eyes, skin sores, and in chronic cases, tissue death, necrosis.
Where does this one pop up?
Fresh and brackish water, reported from maybe 25 countries.
Yeah.
But the trigger conditions vary. Japan, eels in hot weather. Taiwan, eels in cooler spring weather. U.S., often catfish in warmer water with lots of organic matter.
Interesting variation. Then there's E. ictori, enteric septicemia, or hole-in-the-head disease.
Yeah, that descriptive name again. Signs include fish swimming near the surface, obvious external lesions, pale gills, those bulging eyes again, ulcers.
It can cause septicemia.
Yeah, acute blood infection or a more chronic brain inflammation, encephalitis. Doesn't hit as many species as E. tarda, but still quite a few.
And it affects salmonids, too.
Experimentally, yes. Primarily affects channel catfish, though. Spreads when they ingest the bacteria from the water. Fingerlings and adults.
Okay, next. Enteric redmouth disease, or ERM, caused by Yersinia ruckeri.
Another gram-negative one, yeah. An enterobacterium?
The name redmouth gives a clue, I guess?
It certainly does. Key signs. Bulging eyes again, skin darkening, little subcutaneous hemorrhages, bleeds under the skin, especially around the throat and mouth.
Internal signs.
Enlarged spleen, redness, and fluid in the lower intestine. Microscopically, you might see kidney damage.
And this is widespread, especially salmon farms.
Very much so. U.S., Europe, South Africa, Australia. Pretty much global, where salmonids are farmed intensively. It's even found in some aquatic invertebrates.
How does the disease progress?
More acute, faster onset in smaller fish. Can be chronic in larger ones. Yet importantly, survivors can be carriers.
Asymptomatic carriers.
Exactly. They look healthy, but shed the bacteria, infecting others. Stress and poor conditions make outbreaks much worse.
Makes sense. Okay, shifting to Vibrio now. Vibriosis, this is the saltwater one I was thinking of.
Generally, yes, more common in marine or brackish environments, caused by Vibrio genus bacteria. Goes by other names too. Boil disease, saltwater furunculosis, ulcer disease. Gives you an idea of the signs.
What are the main species?
Vibrio angularum causes typical vibriosis, and Vibrio salmonicida causes cold water vibriosis or Hitra disease.
And the signs to watch for?
Lethargy, weight loss, red spots appearing on the body, dark swollen lesions that can burst and ulcerate.
Ulcerate, okay.
Exophthalmia, bulging eyes can happen here too. In acute outbreaks, it spreads fast. Fish might die without showing many signs.
How does it spread?
Horizontally from sick fish. Also through contaminated feed. And again, survivors can be carriers, a reservoir for the bacteria.
All right, that covers a good chunk of the gram-negatives. Let's switch to the gram-positive bacteria now, starting with streptococcosis.
Right. Streptococcosis is a major one, caused mainly by Streptococcus species like S. agalactiae, S. ne, S. discalaxia, and S. ictaluri.
S. ne sounds familiar.
It became a big issue in the 90s, early 2000s, and importantly, it's zoonotic.
Meaning it can infect humans.
Yes. S. ictaluri emerged later, mainly in the U.S. catfish industry. Similar to NEA, but maybe less virulent in channel catfish.
What are the signs of streptococcosis?
Can be subtle at first. Lethargy, maybe loss of appetite, skin might darken.
Sometimes they just die suddenly.
In very acute infections, yes, with few outward signs. But in fish that die or survive, you might see these pus-filled bumps, pustules, often on the jaw or tail.
How does it spread?
Mostly horizontal. But vertical transmission parent to offspring has been reported in tilapia for S. nea and S. agalactiae. The bacteria can survive in water and sediment, too.
Makes it hard to get rid of. Okay, next, gram-positive. Weissellosis. Caused by Weissellocetti sounds newer.
It is, relatively speaking. First found in rainbow trout in China back in 2007. Since then, seen in the U.S., Southeast Brazil. Definitely an emerging issue in farm trout.
Do we know if it affects other fish?
Not clearly yet. Susceptibility of other species is still being looked into.
What triggers it?
High water temperature seems to be a major predisposing factor. The fact it shows up seasonally suggests it can persist in the environment to become endemic.
And it affects larger fish more.
Seems to, yeah.
Yeah.
How it actually transmits is still under investigation.
Interesting. Okay, moving to a more chronic one. Mycobacteriosis, caused by Mycobacterium species. I think I've heard of this in aquariums.
You probably have. It's
Yeah.
caused by species like M. marinum, M. fortuitum, M. cheloni. Tends to be slow but severe.
They're gram-positive, acid-fast rods.
That's right. That acid-fast staining is characteristic. In advanced stages, fish look really wasted, emaciated, bulging eyes, spinal curvature lordosis,
Lordosis.
severe hemorrhages, lethargy, skin lesions, scale loss. It's nasty.
Found everywhere.
Pretty much worldwide, wild and farmed fish, reported in lots of wild species, striped bass, cod, others.
How does it spread? M. marinum?
Not fully understood, but the main suspected route is oral eating infected material or fish.
Then there's no cardiosis, no cardiospecies. Sounds like it causes tissue damage. Granulomas.
Yes, exactly. Caused by N. asteroids and Cereolae, others. It's a lethal infection causing granulomas, those walled-off inflammatory lumps in muscle, skin, internal organs.
What are the visible signs?
Nodules in organs, sometimes with skin ulcers breaking out, lethargy, anorexia, cloudy eyes, bleeding in or around the eye.
Where is it a big problem?
Causes major economic losses, especially in Asian aquaculture. And Cereolae has hit Japanese sea bass, yellow croaker hard in China and Taiwan. High mortality.
Transmission.
Still not entirely clear. Maybe oral route, possibly contaminated feed. Moving fish around might spread it. Vertical transmission is suspected in barramundi. Even amoebae are being looked at as potential carriers.
Amoebae. Wow. Okay, last one in this section. Franciscellosis, caused by Francisella, another recent one.
Relatively, yeah.
Yeah.
Documented more recently. Caused by F. notuensis, F. pituicida, F. victoria. Quick correction here from the outline. These are actually gram-negative.
Ah, okay. Gram-negative coccobacilli.
Yes. Facultative intracellular non-modal shape can vary. Signs include exophthalmia, loss of appetite, lethargy, little pinpoint bleeds called petechiae,
Petechiae, right.
abnormal swimming, dark body color, hemorrhagic nodules, those blood-filled lumps,
Found in many places.
things like it, tilapia across the Americas, Taiwan, Hawaii, also three-line grunts in Japan, cod in Norway, Atlantic salmon in Chile. Pretty global distribution, broad host range.
Life cycle known.
Not fully. Probably similar to the mammalian Francella. Spreads horizontally in tilapia, likely through the water. Different strains might behave differently. Role of vectors or vertical transmission. Still unknown.
Wow, okay. That is a lot of bacterial threats. Let's pivot now to what we can actually do about them. Prevention is key, right?
Absolutely. Always better to prevent than treat. Less costly, better for the fish, avoids issues like resistance.
So what are the standard sort of conventional prevention methods?
Good basic hygiene is fundamental. Using pathogen-free broodstock, screening new fish, sterilizing equipment, culling, removing infected fish quickly.
Are there movement restrictions?
Yes, for some serious ones like BKD, furanculosis, ERM, there are often rules about moving fish to stop the spread.
What else? Quarantine.
Quarantine is vital for new arrivals. Maintaining good water quality, good nutrition, sometimes adding supplements like vitamins A, E, C, can help against certain bacteria like Aromonas hydrophila.
Boosting immunity.
Exactly. And strict biosecurity, preventing pathogens coming in via equipment, visitors, staff. Better hatchery management helps against Aromonas. Using ozone or UV in water reuse systems is good too.
And disinfectants.
Yeah, they play a role. Various types are used chlorine-based, quaternary ammonium compounds, iodophores, depending on the situation.
Then we get to antibiotics. Effective, but problems.
Yeah, big problems. Yes, they can work, but overuse leads to antimicrobial resistance. That's a massive concern for fish health and human health. Plus, you have issues with drug residues in food.
So the industry is looking for alternatives.
Actively seeking alternatives, yes.
Which leads us to some of the newer approaches. Probiotics.
Right, live or dead microbes, or their components. They work in various ways, competing with pathogens, improving water quality, stimulating immunity.
How are they chosen?
Often based on lab tests, seeing if they can kill or inhibit the target pathogen. Can be added to feed or water. Liquid forms might act faster.
Any examples?
Yeah. Certain bacillus species, like Subtilis and Lichenoformis, have shown promise protecting trout against Yersinia ruckeri.
Okay. And prebiotics, how are they different?
Prebiotics aren't live microbes. They're non-digestible feed ingredients that selectively feed the beneficial bacteria already in the fish's gut.
Like fertilizer for good gut bugs.
Kind of, yeah. Mannan oligosaccharides, MOS, are one type, showing good results for immunity and growth. Beta-glucans are another, acting as immunostimulants.
Nanotechnology is even playing a role.
It is, surprisingly, using nanoparticles to deliver drugs more effectively or to enhance vaccines, nanovaccines.
How does that help?
The idea is targeted delivery, protecting the drug or antigen, improving absorption, maybe reducing side effects. It's still developing, but promising.
And bacteriophage therapy, using viruses against bacteria.
Yeah, it sounds futuristic, but it's a real area of research. Phages are viruses that only infect specific bacteria. They're harmless to animals and humans.
So you could use them for control?
Potentially for prevention, treatment, even rapid detection. They're highly specific. Phages have been tried against A. hydrophila, F. saccharophilum in ponds. Some suggest using phage cocktails against things like A. salmonella.
Cocktails, multiple phages.
Yeah, to improve effectiveness and reduce the chance of resistance developing. Getting the right phages and doses is critical, though.
And finally, vaccination, a standard tool in animal health.
Absolutely. Traditional fish vaccines often use killed bacteria bacterins inactivated with formalin.
Downsides.
Well, vaccinated fish might still carry the bacteria asymptomatically, and traditional injection methods are stressful and often give shorter-term protection. So the industry is always looking for better, easier, more effective vaccination methods.
Okay, that's a really thorough look at the bacterial side. Let's switch gears completely now to the parasite. Marine white spot disease. Cryptocaryon irritans,
Right, the saltwater equivalent of freshwater eek, essentially.
caused by a ciliated protozoan.
Exactly, a single-celled parasite. Causes marine eek, or marine white spot. Affects marine fish, wild and farmed, in warmer waters, roughly 15 to 30 degrees C.
A big headache for aquarists and fish farmers.
Huge problem worldwide. Infects a lot of different fish species, though susceptibility varies.
And like the bacteria, there are different strains.
Yeah, studies show genetic differences globally. These can affect the life cycle duration, tolerance to salinity changes.
So maybe they're different species.
Some researchers suggest that possibility, but for now, it's all considered Cryptocaryon irritans. The signs, the complex life cycle, the speed it can kill often within days are similar to freshwater yeast, if the obvious,
But they're not closely related.
only distantly. Big differences in salinity tolerance and life cycle length. Accurate diagnosis and rapid response are absolutely crucial.
Okay, so what are those key signs? The white spots.
That's the classic sign, yes. Small white spots, nodules, patches on fins, skin gills, often visible to the naked eye.
What else?
Ragged fins, cloudy eyes, pale gills, maybe excess mucus, skin color changes, looking thin.
But you might not always see the spots.
Crucially, no,
Yeah.
especially on pale fish or if it's mainly in the gills. So, absence of spots doesn't mean no infection. Always check the gills if you suspect it.
Behavioral signs.
Flashing, scratching against things, abnormal swimming, hanging at the surface or bottom, lethargy, rapid breathing indicating distress.
And rapid die-offs in a population.
Yes, often a sharp increase in mortality over just a few days. How bad it gets depends on the parasite strain, fish species, previous exposure, water temp, lots of factors.
Let's get into that life cycle. Direct but complex stages on and off the fish.
Correct. No intermediate host needed, but several stages. Starts with the trophont, that's the feeding stage, embedded in the fish tissue skin or gills.
What does it look like?
Spherical to pear-shaped, covered in cilia, kind of rolls around under the epithelium. It's fairly protected from treatments at this stage. Feeds on fluids and cells for 3 to 7 days. Then leaves the host.
It leaves even if the fish dies.
Yes, but it's not immediately infective. It needs to develop further.
Okay, so after leaving the fish,
It becomes a protomont. Loses cilia, flattens out, moves on the substrate for a few hours, maybe 2 to 18 hours. It might be briefly susceptible to treatment here.
then it encysts.
Right. It stops, sticks to a surface, forms a cyst, becomes a tumont. This is the reproductive stage. The cyst hardens over 8-12 hours, giving it great protection. Inside, it divides into hundreds of daughter cells called tumites.
Tumites. And they get released.
Exactly. They break out of the cyst as the tumonts. Free swimming, infective stage, actively seeking a new fish host.
And the whole cycle time depends on temperature.
Heavily. Can be 6-11 days at 27 degrees C, or over 5 weeks at lower temps. The tunes only survive 12-24 hours without a host in warm water, maybe longer if it's cooler. But one tumont can release hundreds of those tumonts.
That's a lot of potential infections. Can they go dormant?
There's evidence one strain's trophonts and tormonts survived dormant at 12 degrees C for months, then reactivated when the temp went up. So yes, persistence is a problem.
Very adaptable. Can fish develop immunity?
They can, yes. Fish surviving an infection can have immunity for up to 6 months.
But they can still be carriers.
Unfortunately, yes. Survivors can be reservoirs for future outbreaks. Vaccine development has been ongoing for years, some promising early results, but nothing commercial yet.
And general fish health matters?
Absolutely. Good health, good water quality helps the immune system fight back. Compromised fish suffer much more severe infections.
Diagnosis sounds tricky if you can't always see the spots.
It can be. White spots resemble other things sometimes. Definitive diagnosis needs microscopy.
Looking at skin, fin, gill samples.
Yes. You need to see the parasite, the trophonts rolling around, or other stages, and distinguish it from freshwater based on morphology, size, the macronucleus shape. You have to visualize the parasite.
So treatment, what works? What are we targeting?
Primarily the free swimming, the free colts, maybe the tomons less effectively. The trophons on the fish are pretty protected.
Copper sulfate. Is that still the main one?
Historically, yes, often the most common and effective. But dosage is critical. Depends on the fish species, invertebrates present, water chemistry.
Need to test the water often.
Essential to maintain therapeutic levels, typically 0.15, 0.30 ppm, without reaching toxic levels.
What about chelated copper?
More stable, supposedly,
Yeah.
but safety and effectiveness can vary. Always follow manufacturer's instructions carefully.
Other options? Formalin.
Formalin can work against serrans and defaultants, especially at higher doses or longer baths, but it can deplete oxygen, so you need good aeration.
Chloroquine.
Chloroquine phosphate is used in some systems, yeah. Often a prolonged bath for several days.
What about lowering salinity? Hyposalinity.
That aims to disrupt the parasite's internal water balance, especially the serrans, lowering to 10-15 parts per thousand, but effectiveness really varies.
Why does it vary?
Some Cryptocaryon strains tolerate low salinity much better than others. Some outbreaks have involved strains happy down to 5 or 10 ppt, so it might not work.
So it's not a guaranteed fix.
Definitely not. If you try it, the suggestion is often 15 ppt for 3-4 weeks, lowering salinity gradually,
Raising temperature.
can speed up the life cycle, potentially shortening treatment time.
Oh.
But it also stresses the fish, so it's the trade-off.
What about just moving the fish?
Transferring fish to a clean tank every, say, 3 days can break the cycle as trophons leave. But the original tank must stay empty of fish fallow for at least 4 weeks, maybe longer in cool water, to ensure all tomons die off.
Anything else? Ozone. UV.
Ozone and UV sterilization can kill the tomons in the water being treated.
And disinfecting equipment is crucial.
Absolutely vital to kill any lingering tomons on nets, tanks, pipes, everything.
Can you give examples of disinfectants for Cryptocaryon?
Sure. Benzalkonium chloride, 100 mg for an hour, kills tomons and the tomons. Chlorine needs higher levels for tomons, maybe 60 mg for 24, and the tomonons, 2.4 mg for 1 hour.
What about non-chemical methods?
Complete drying for 24 hours is effective. Heating equipment to 40 degrees C for an hour also works against both stages.
Okay. Given all this, biosecurity must be paramount.
Absolutely critical.
Yeah.
Quarantine is non-negotiable for new fish. And not just fish, marine plants, invertebrates, substrate, anything that get carried in months needs quarantine too.
Work with a specialist on that.
Highly recommended. They can help design a robust quarantine and biosecurity plan. Disinfect all used gear. Be aware of potential aerosol spread between tanks, and check your source water. Raw seawater can be a reservoir.
So, summing up treatment, copper is still a go-to, but strain variation matters. Hyposalinity is unreliable. Quarantine and biosecurity are key.
That captures it well. Copper sulfate pentahydrate seems most consistently effective. Chloroquine, formalin, tavoyl. But yeah, low salinity tolerant strains make hyposalinity tricky, and you absolutely need that quarantine and source water management. Dormant termomes are a hidden threat. A fish health pro is your best ally here.
Well, this has truly been a deep dive. Bacterial culprits, this tricky parasite. It's clear there's a huge range of diseases impacting fish.
Both farmed and wild populations are vulnerable, yeah. And understanding the specifics, causes, signs, spread, control is just essential to manage the economic and ecological fallout.
And it seems like things are always changing, new pathogens emerging, highlights the need for ongoing research, better diagnostics.
Couldn't agree more. Which brings us to a final thought for you, the listener. You know, considering how connected aquatic ecosystems are, the global trade in fish, how can we all, individuals and industries, contribute more effectively to preventing disease spread?
And ensuring fish health worldwide.
Exactly.
Right.
What's our responsibility to support sustainable aquaculture, to invest in the research needed for new control methods? It's a complex global challenge that needs real commitment.